Hello, everyone, and welcome to today's webinar, Spatial Protein and RNA Profiling, Seeing the Unseen in the Tumor Microenvironment. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. To learn more, visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Jonathan Nowak, molecular and gastrointestinal pathologist at Brigham and Women's Hospital and the Dana-Farber Cancer Institute, assistant professor of pathology at Harvard Medical School. For a complete biography on our speaker, please click on the presenter tab. Dr. Nowak, thank you for joining us. You may now begin your presentation.
I'm Jonathan Nowak from the Dana-Farber Cancer Institute, and it's my pleasure to spend the next hour telling you about how my lab uses spatial profiling to see the unseen in the tumor microenvironment. My lab studies gastrointestinal cancer, particularly colorectal and pancreatic cancer, and it's the latter that I'll focus on today. Pancreatic cancer is one of the most lethal solid tumors. It's the third most common cause of cancer-related mortality in men and women, almost exactly on par with colon cancer. One of the reasons for this is that about 80% of patients present with late-stage disease where a potentially curative resection is not possible. Even for those that present with stage 1 or 2 disease, as you can see on the left, 5-year survival is 50% or less. Most of these patients, unfortunately, will eventually recur and die of their disease. The lab that I run jointly with Brian Wolpin at the Dana-Farber addresses this situation in two different ways. First, we work to understand treatment response and resistance mechanisms in patients with established pancreatic cancer. Second, we work on ways to find pancreatic cancer earlier when there's a greater chance for a cure. For today's talk, I'd like to touch on 3 different topics. First, I'll go over our tissue-based approach for studying GI cancer. Next, we'll look at some examples of how we identified targetable pathways that regulate the GI cancer microenvironment using both large landscape studies and clinical trial correlative studies. And then finally, we'll talk about how human and mouse studies can be integrated to functionally explore hypotheses that we derive from human specimens. Like most solid tumors, there are now some genomic biomarkers that can be used for therapy selection in pancreatic cancer, as you can see here. However, most of these occur really quite infrequently. And although tumors that are deficient in homologous recombination represent the largest potential class of targetable tumors, these tumors are often difficult to detect using clinical sequencing approaches. Overall, about 15% or less of pancreatic cancers we see in routine practice have a potentially targetable alteration. And in addition, there's been really very little success with immunotherapy for pancreatic cancer, so there's no role for PD-L1 testing. And it's clear that we need to look beyond genomics in order to identify better ways to treat pancreatic cancer. So if we're going to study pancreatic cancer tissue, what cell types should we go after? And as this cartoon illustrates, there are really many possible and intriguing options. Tumor cells are certainly important, as shown here in red. But also important are the immune cells that are present in the tumor microenvironment. This can be pro-tumorigenic immune cells and also anti-tumorigenic immune cells. Beyond that, fibroblasts, also a very important and maybe dominant component for most pancreatic cancers. And there are additional other stromal cell types like endothelial cells and nerve cells that probably also are important in orchestrating how a tumor behaves. For some of these cell types, we can recognize them quite nicely using standard histologic approaches. And our lab has done some work using traditional H&E-based stains to identify different immune cell populations in the tumor microenvironment. This is an example of one study we did a number of years ago looking at immune cells in a cohort of approximately 1,000 colorectal cancer patients and outlining the image analysis approach that we used to identify 4 different types of immune cells in colorectal cancer. While not all immune cells can be identified by morphologic analysis alone, some immune cell types can. These include lymphocytes, plasma cells, neutrophils, and eosinophils. We trained machine learning tools to identify these cells just by using H&E morphology and showed that they performed quite well in comparison to manual identification by a GI pathologist. We then used these tools to analyze our cohort of colorectal cancer patients. What we found is that immune cells tend to occur in different regions of the tissue preferentially. And the plot on the left shows densities in intraepithelial versus stromal areas. And for all four immune cell types that we're able to identify, we found densities that were significantly higher in stromal areas compared to intraepithelial areas. When we then looked at stromal density, we found that across all four of these immune cell populations, tumors with higher levels of either lymphocytes, plasma cells, neutrophils, or eosinophils were all associated with better colorectal cancer specific survival. This had already been shown for lymphocytes, but was really unclear or much less well known for plasma cells, neutrophils, and eosinophils, suggesting that these other less common immune cell types are also potentially playing an important role in the pancreatic cancer immune response. In addition to simply counting how many cells were present, we could also take advantage of the fact that we knew where each of these immune cells was located. And there are a number of ways that we can use this data to analyze the degree of colocalization between tumor cells and immune cells. One of the most common ways we do this is using a simple mathematical function called the G-cross function. And in short, this function allows us to estimate the likelihood of any tumor cell in the sample having at least one neighboring immune cell within a radius that is pre-specified for the analysis. You can plot the output of this function as a curve and then look at the area under the curve. And you can see that with a higher area under the curve, you are able to identify tumors with greater colocalization of immune cells with tumor cells, as you can see on the right in panel C, as compared to panel B in the middle of the slide. When we applied the G-cross function to look at the spatial organization of our four immune cell populations, we found that for lymphocytes and for neutrophils, greater colocalization between tumor cells and the respective immune cell type was associated with significantly better colorectal cancer specific survival, as you can see looking at category 1 through category 4 in both of those plots. In contrast, the spatial organization of plasma cells and eosinophils does not really seem to affect patient survival at all. We can look at these results and then start to make hypotheses about why it's important for lymphocytes and neutrophils to be located nearer to tumor cells, whereas whatever the function of plasma cells and eosinophils might be doing is something that is not necessarily requiring co-location of tumor cells and the respective immune cells. So although we can see some things by H&E analysis, the reality is that most cell types are not really identifiable just by looking at H&E slides. And to identify these cell types, we need RNA markers, we need protein markers, and we need to be able to combine those markers to identify all of the other elements in the tumor microenvironment. And a major focus of our lab is building assays that allow us to do this. This includes multispectral immunofluorescence, spatial transcriptomics, and in situ mutation detection. We can then apply these assays to tissue and use the output digital images as input into machine learning tools and automated analysis pipelines that allow us to go from simply qualitatively seeing a tissue, as you can see on the left, to measuring it and knowing the identities and the locations of all of the different cells that make up the tumor microenvironment. And one of the tools that's really the linchpin for this workflow in the lab is our LicaBond autostainer. This is the input to almost all of our assays, regardless of whether they're protein-based immunofluorescence, spatial transcriptional profiling, or in situ mutation detection, or even in some cases, classical immunohistochemistry. As many of you know, multiple assay types are available for protein-based tissue imaging. We can think of these along two major axes of differentiation, one of which is plexing, or the number of markers that can be included in each assay, and the other is the tissue area throughput. Traditional single-color immunohistochemistry, or H&E staining, offers very high throughput, but has a very limited plexing capacity. Conventional immunofluorescence can look at 3 or 4 markers together potentially, slightly higher plexing, slightly lower throughput. Multispectral immunofluorescence can look at up to 9 markers and offers a really nice balance of plex and throughput. And then there are multiple additional technologies, including multi-round immunohistochemistry and immunofluorescence, DNA barcoded antibody approaches, and tissue cytoph and MIBI that offer the opportunity to look at dozens of markers, but at the cost of having a very low tissue throughput. We found for most of the assays in our lab that multispectral immunofluorescence offers the best trade-off between plexing and tissue throughput. But I think it's important to note that all of these approaches are only as good as the antibodies or stains that underlie them, and that it's very important to choose the technology that helps you answer your particular scientific question. And all of these questions require a balance of throughput, plexing, and cost. Regardless of the wet lab assay that you choose to run for your study, it's also very important to have an appropriate image analysis pipeline in place. This is an image from one of the very first multiplex immunofluorescence assays that we built in my lab. It's a very simple image that shows cytokeratin positive tumor cells and CD8 positive T cells in purple. As you can see, there's probably about 5 different cells that we would consider T cells here. Some of those are within intraepithelial areas or surrounded by tumor cells, and there are a couple T cells in the stroma as well. And if we decided to just write a rule that explicitly said identify T cells, you might look for circumferential expression of CD8. And as you can see here, really none of these cells actually have a circumferential CD8 expression pattern in the membrane, but they are all cells that we would probably want to recognize as T cells. And being able to do this sort of analysis appropriately often requires supervised machine learning. And this allows you to look at many different features of these cells, including not just the expression of one single marker to define them as T cells. And so this is really a backbone of the work that our lab does. We really like this approach because it allows for a high level of concordance with a pathologist interpretation as a gold standard. And it also allows us to do tissue segmentation to identify different regions of tissue and to identify specific cell types. If you're unsatisfied with how your algorithm is working, you can continue to train it, or you can train a new algorithm until you get to an acceptable level of performance. Importantly, these algorithms are also human interpretable, so if they're not working correctly, you can look under the hood and understand what they're doing that is not in accordance with what you think they should be using to classify cells. These tools are also able to consider both cell morphology and marker expression. And importantly, these assignments, whether it's a tissue region or a cell phenotype, come with a confidence score so that you can exclude, if you would like to, low confidence calls from the algorithms. This slide shows an example of an analysis that we've done across more than 2,500 colorectal cancers across 4 large North American cohorts. All of the tissue that was in these studies came from different institutions, was prepared at different times, and has been stored under very different conditions. You can see the ages and the specifics for the different cohorts shown on the right. However, although you might expect differences in antigenicity to give you very different tissue profiles across these tumors, when we used supervised machine learning to analyze our multiplex immunofluorescence data, we were able to show that densities of T cells, whether they're CD4 positive or CD8 positive T cells, were nearly identical across these cohorts, even though the underlying quality of the tissue is actually quite different. And I think this is just a very nice example of the power of machine learning in allowing you to generate harmonized data that can be compared across large numbers of cases and even multiple cohorts that have been built under very different conditions. So for the next part of my talk, I'd like to show a couple examples of how we've identified targetable pathways that regulate the microenvironment of pancreatic cancer. When we think about pancreatic cancer, there are many possible cells that would be interesting and worthwhile to study. Most obviously, the tumor cells themselves. In pancreatic cancer, there are 2 major transcriptional subtypes of tumor cells that we recognize, classical cells and basal cells. In addition to that, the pancreatic cancer microenvironment is characterized by a very abundant fibroblast population. We know these fibroblasts can move between an activated and a more quiescent state. And then beyond tumor cells and fibroblasts, we know that there are many immune cells in pancreatic cancer, but in aggregate, they seem to have more of an immunosuppressive phenotype that tends to promote tumor growth. One of the first cell populations that we wanted to study in pancreatic cancer were myeloid cells. We built a number of multiplex immunofluorescence assays to do this, including one that looked at macrophages and their degree of polarization. We used this assay to study about 300 primary resected tumors and found that, surprisingly, macrophage density harbored very little prognostic significance. And this was surprising because macrophages are actually the dominant immune cell population in most pancreatic cancers. While we were doing this work, we made the interesting observation that macrophages aren't distributed randomly in the tumor microenvironment. Rather, M1 macrophages are overall closer to tumor cells than M2 macrophages, as you can see in the lower left. And when we then looked at proximity and survival, we found that tumors with immunosuppressive M2-like macrophages closer to tumor cells were associated with worse survival, as shown in the plot at the lower right. And this was really true across a range of distances. And in contrast, M1 macrophage location did not associate with survival. This showed us that it's not just how many cells are present in the immune microenvironment, but it's actually where those cells are located that's important. More recently, patients with pancreatic cancer are typically treated with neoadjuvant chemotherapy and radiation prior to having their tumor resected. So what we really wanted to know was how this therapy is changing the immune microenvironment in pancreatic cancer. And to do this, one of the other postdocs in my lab conducted a study where she assembled cohorts of patients that had upfront surgery and also patients that got neoadjuvant therapy. And then we analyzed these with multiple immunofluorescence panels that allowed us to study T cell subsets, macrophage polarization, as well as myeloid cell subsets. We took that data and used it to ask multiple questions about the organization of immune cells. We were able to look at density, of course, but we could also look at compartment. Are the immune cells within the tumor intraepithelial area, or are they localized in the stroma? We're also able to look at multiple measures of proximity between tumor cells and immune cells, and we're also able to look at heterogeneity across the tumor. Are immune cell populations different in the center of the tumor versus the periphery? Here's the high-level view of those main immune cell densities across our 300 tumor primary resection cohort. As you can see, the total density of immune cells is really quite variable across the cohort, but when you look at the individual immune cell populations in the tracks at the bottom, it's clear that there's no single immune cell population that's really driving these density differences. When we then looked at these densities in terms of whether the immune cells were preferentially located in the intraepithelial or stromal regions, we found that some immune cells, like T cells, are much more likely to be enriched in stroma. In contrast, granulocytes, such as CD15 positive cells at the bottom, are distributed much more equally between intraepithelial and stromal areas. We were also able to look at the proximity of various immune cell populations to tumor cells. And in this plot, you can see at the top that either CD4 or CD8 positive T cells tend to be the furthest away from tumor cells. And interestingly, CD4s are a little bit further away than CD8s. Macrophages come in the middle, and as we saw previously, M1 macrophages are a little bit closer to tumor cells than M2 macrophages. And then finally, CD15 positive granulocytes tend to be closer to tumor cells than either macrophages or T cells. We next asked whether we could find recurrent patterns of immune cell infiltration across different tumors. The unsupervised clustering shown here identified 4 major patterns or classes of tumors. 2 of these were dominated by ARG1 positive immunosuppressive granulocytes and were further differentiated by M2 versus M1 macrophage predominance. An additional cluster, C3, was characterized by somewhat elevated T cell densities and higher M2 macrophage densities, while C4 was dominated by T cells. Notably, none of the patterns correlated with the status of key genomic drivers, nor did they associate with tumor mutational burden. we hypothesized that tumors in the C4 cluster, which had the highest T cell densities and the lowest densities of immunosuppressive granulocytes or M2 macrophages, would be associated with better survival. And this turned out to be true, as shown in the Kaplan-Meier plot comparing C4 to a C1 to C3 combined group. In contrast to other tumor types, such as melanoma and colon cancer, where densities of individual immune cells have been strongly proven to harbor prognostic significance, studies of pancreatic cancer have been much more variable, with comparatively less consensus about individual cell populations. Some studies say T cells don't matter, some say CD3, CD8s matter, while others say that it's the helper T cells that are actually the most important. One reason for these disparate findings may be that it's not the absolute level of individual immune cell populations that matters, but rather the relative abundance of different populations that may have opposing functions. Having documented our findings in tumors from upfront resections, we turn to neoadjuvant treated cases. To control for confounding due to variable tumor response to treatment, we only studied cases where all tumor and slides were available for review, allowing us to both uniformly grade tumor response and to select the most representative blocks from neoadjuvant treated cases for analysis. As you can see in the plots here, these two cohorts look pretty similar at first glance, and we were really quite surprised to find no significant difference in total immune cell density between upfront surgery and neoadjuvant treated cases, as shown on the right. We therefore studied immune cell substance in greater detail. Just as we found for total immune cell density, CD3 density wasn't different. However, delving in deeper, we found a significant increase in CD3-CD8 density and a more modest decrease in CD3-CD4 density. Together, these changes resulted in a significantly decreased CD4 to CD8 ratio in neoadjuvant treated cases. This can be appreciated visually in the MIF images and phenoplots at the top, where the overall T cell balance shifts from CD4 dominated to CD8 dominated, or cyan to pink. Similarly, when we looked at CD15 positive granulocytes, we found no significant difference between upfront surgery and neoadjuvant treatment. However, the density of ARG1 positive immunosuppressive granulocytes was significantly decreased in neoadjuvant cases, as you can see in the top right, by a reduction in red double positive cells. Although these cells are fairly rare overall, our prior work has shown that they are one of the strongest immune predictors of outcome for primary pancreatic cancer patients. Finally, for macrophages, we also found that overall density did not change. However, there was a significant increase in M1 polarized macrophages and a slight decrease in M2 polarized macrophages. Overall, this resulted in a significant shift in the M1 to M2 ratio, with neoadjuvant tumors moving towards an anti-tumorigenic M1 dominated microenvironment, as indicated by cyan cells in the phenoplot in the upper right. Overall, these results indicated that neoadjuvant therapy wasn't associated with a change in total immune cell density, but was associated with multiple shifts in the immune cell subpopulations that all tilted towards a more anti-tumorigenic response. To see if this was true, we did an analysis where we reclustered our upfront surgery cohort with a neoadjuvant cohort and mapped back to the 4 main clusters we initially identified. We found that although the T-cell rich cluster was one of the smaller clusters in the upfront surgery cohort, more than 2/3 of all neoadjuvant treated cases mapped to this cluster. While we would need paired pre- and post-treatment specimens to formally ascribe this shift to neoadjuvant therapy itself, it does suggest that neoadjuvant therapy alters the tumor microenvironment in a favorable manner. We next looked at the proximity of stromal macrophages to tumor cells in neoadjuvant treated tumors. Using multiple measures of proximity, we found that M1 polarized macrophages, but not M2 polarized macrophages, were closer to tumor cells in neoadjuvant versus upfront surgery cases, and results for the G-cross analysis are shown here. These results suggested that the immune microenvironment of neoadjuvant treated pancreatic cancer doesn't just represent the immune cells that are left over once tumor glands die and disappear, but rather that the organization of immune cells in the microenvironment actively changes, with M1 macrophages seeking out the remaining tumor glands, getting closer to them, and even becoming intraepithelial, all while these glands are shrinking and disappearing from the tumor microenvironment. These results prompted us to next look at macrophage configuration and histologic response in neoadjuvant treated tumors. For this purpose, we divided tumors into good responders and poor responders based on the modified Ryan scheme and the CAP reporting protocol. Good responders had a grade 1 score, while poor responders were grade 2 or 3. And some examples of both of these are shown here with the corresponding macrophage data. We knew that neoadjuvant therapy overall was associated with higher intraepithelial macrophage density. And stratifying this association by response showed that tumors with a better histologic response had a greater increase in total intraepithelial macrophages as compared to poor responders. And this was driven entirely by M1 polarized macrophages. Although our total case numbers were modest, we did find that within neoadjuvant treated tumors, higher intraepithelial M1 polarized macrophage density was associated with better overall survival. Analysis of macrophage proximity to tumor cells showed similar results. Tumors with a good histologic response exhibited greater proximity of M1 macrophages to the few tumor glands that remained, as opposed to tumors with a poor histologic response, where tumor glands were more numerous and would theoretically offer a greater opportunity for close proximity to M1 macrophages just by chance. Finally, when we looked at survival in neoadjuvant treated patients, we found that greater proximity between M1 macrophages and tumor cells was associated with significantly better survival. Interestingly, this association was different than what we had observed in upfront resected cancer, where greater M2 macrophage proximity to tumor cells was associated with worse survival. This suggests that the immune microenvironment shifts in neoadjuvant treated cases are functionally relevant and that these tumors may be immunologically distinct. Although these results were very interesting, they all represented just a static snapshot in time, and they couldn't really tell us what was actually organizing and regulating macrophage spatial organization. So to determine what signals actually regulate this, we turned to the COSMIC spatial molecular imaging platform and profiled more than 60 regions of normal pancreas, pancreatic intraepithelial neoplasia, and invasive pancreatic cancer from our primary resected specimens. Here's a clustering analysis of all 250,000 cells that we initially identified. Many different cell types were identified here, including macrophages and 2 tumor epithelial subtypes, which we found correlated with known basal and classical subtypes of pancreatic cancer. Because the COSMIC system also provides immunofluorescence data for 4 protein markers, as you can see here on the right, we're able to perform orthogonal sanity checks in the RNA data and confirm, for example, that cytokeratin expression at the protein level was restricted to tumor cells and pancreatic ductal cells, whereas CD45 protein expression was restricted to immune cells. Here's a very zoomed-in view of a single pancreatic islet showing the cell segmentation and inferred cell identities with the color key on the right. Overlaid on this image are dots indicating RNA hybridization signals for 5 of the 1,000 profiled genes, including REG1A, which is an acinar cell marker, and insulin and secretogranin 5, which are markers of beta cells and delta cells, respectively. These can be easily distinguished in the islet, which is largely surrounded by a mixture of yellow acinar cells and blue ductal cells. So we first wanted to know whether the macrophage localization differences that we observed by protein analysis in MIF were picked up by RNA analysis. When we looked at RNA for the same M2 polarized macrophage markers that were included in our MIF panel, we saw a very nice trend for increased expression of these markers in macrophages that were further away from tumor cells. This is very reassuring, showing that the RNA-based assay could recapitulate what we had previously seen in protein space. We then asked what other genes are differentially expressed in macrophages based on distance from tumor cells in a pattern that matches M1 versus M2 polarization. We are excited to find 16 additional genes, many of which regulate cell function and cytokine signaling. These represent promising candidates for identifying the mechanisms that specify macrophage spatial organization in the tumor microenvironment. For example, could we manipulate CCL2, CXCL14, or IL-1 signaling to push M2 macrophages further away from tumor cells? We're currently pursuing functional studies to test these hypotheses and others using model systems. and had we not been able to transcriptionally profile individual macrophages in a spatially resolved manner, it would have been nearly impossible to make these observations. Next, I'd like to share an example of how we used our spatial profiling approaches to understand pancreatic cancer biology in the context of clinical trials. Going back to the pancreatic cancer microenvironment, I'd like to remind you that fibroblasts are one of the most dominant cell types in pancreatic cancer and are responsible for the dense stroma that surrounds tumor cells. To understand fibroblasts better, we built a multiplex immunofluorescence assay that allowed us to identify different populations of cells, including fibroblasts, immune cells, stromal cells, and tumor cells in the microenvironment. We ran our panel across our group of about 300 primary resected tumors, and as the plot shows here, you can see that fibroblasts are the dominant cell type in most tumors, comprising more than a third of the total cellular population within the pancreatic cancer microenvironment. Why fibroblasts are so abundant in the pancreatic cancer microenvironment and what controls their activities has been a long-standing question in the field. Several lines of evidence have suggested that vitamin D signaling is a major regulator of fibroblast activity. Observational studies have shown that patients with higher levels of serum vitamin D who have pancreatic cancer have better outcomes. In addition, we know that tumors that express higher levels of VDR, the vitamin D nuclear receptor, are also associated with better outcomes. And a number of mouse models have shown that vitamin D is key for promoting fibroblast quiescence within the stroma surrounding pancreatic cancer. However, the story is a little bit more complex than that because vitamin D also appears to augment tumor cell classical basal subtypes by pushing cells to more extreme polarization states. In addition, there's a large body of literature that suggests that vitamin D can also modulate immune cell activity, potentially by reducing myeloid-derived suppressor cell activity and modulating T-cell effector function, and maybe also altering immune checkpoint status. And as an aside, worthwhile to note here that although we often think of vitamin D signaling as involved in calcium homeostasis, this signaling pathway actually evolved before the existence of organisms with a skeleton. And it's thought that vitamin D really started out by regulating energy metabolism and then was co-opted for regulating adaptive immunity before it ever became involved in calcium homeostasis. And so based on these initial lines of evidence that suggested that altering vitamin D signaling might be able to alter fibroblast activity in pancreatic cancer, the DFCI, working with the University of Pennsylvania, conducted this 3-armed clinical trial testing the vitamin D receptor agonist paracalcitol along with a standard of care chemotherapeutic regimen in patients with metastatic pancreatic cancer. In this study, patients were randomized to one of 3 arms, receiving either chemotherapy alone or intravenous paracalcitol 1 time per week or oral paracalcitol. And very importantly in this study, patients had a pre-treatment tumor biopsy and then also had a second paired biopsy after 6 to 8 weeks on therapy. This slide is an example of the types of correlative studies that we can do from core biopsies on these trials. By taking multiple cores from a core needle biopsy of metastasis, we're able to perform multiple studies, including single-cell or single-nucleus RNA sequencing, bulk DNA sequencing if needed, and also spatial profiling such as multiplex immunofluorescence and spatial transcriptional profiling. When possible, we also dissociate a core and try to derive organoid cultures that allow us to then study how that tumor responds in vitro. And this is an example of really one of the main reasons why spatial approaches are very helpful for core biopsy specimens. This is a standard liver core biopsy from one of the patients on our trial. It's about 2 centimeters long, and I've highlighted 4 areas here. And as you may be able to tell, none of these areas look histologically normal, but only one of these, which is number 3, actually contains invasive cancer. And so if you were to take this entire specimen, dissociate it, and perform single-cell or single-nucleus RNA sequencing, you'd spend a lot of time studying cells that are really not associated with the cancer. This is especially true for immune cells and stromal cells, where if you were studying the cells that came from region 1, these are more than half a centimeter away
from where the actual tumor is located and are quite likely not directly involved in the response to the tumor or its actual microenvironment. One of the first questions that we wanted to answer for tissue from this trial is whether the vitamin D receptor is actually expressed and which cells are expressing it. We used a multiplex immunofluorescence assay to do this, looking at VDR expression and CYP27B1, which is one of the major enzymes in the activation cascade for vitamin D signaling, and also using a panel of lineage markers that were able to determine whether the vitamin D receptor was expressed within tumor cells, within stromal cells, or within several immune cell populations. And as usual, we analyzed these data using multispectral digital imaging and supervised machine learning, and then we're able to generate plots like the one shown on the right here. When we ran this assay across tissue from our patients in the study, these are the results that we found. And so each of these stacked bar plots represents a separate patient, and they're broken out by arms. And the coloring represents the proportion of cells that are either tumor cells or stromal cells or immune cells that are positive or negative for vitamin D receptor expression. And as you can see, each of these stacked bars really looks quite different from one to the other. So there's a lot of heterogeneity here in terms of the relative expression of VDR positive and negative tumor, stromal, and immune cells. The other point that's worth mentioning here is that in almost all of these tumors, there are populations of each of those main cell types that are positive for vitamin D receptor expression. And so this really suggests that if you're going to understand this signaling pathway within the tumor microenvironment, you need to pay attention to each of these three different cell types because they could all quite likely be responding to vitamin D agonism. With that data in mind, we wanted to find a way to study vitamin D signaling across these three cell populations in an unbiased fashion. And to do that, we turned to the GeoMx Digital Spatial Profiler platform, which allows you to perform whole transcriptome sequencing in selected user-definable areas of tissue. And for the assay we built, we used antibodies against cytokeratin and CD45 and developed a real-time image segmentation tool that allowed us to specifically highlight areas of tumor epithelium as well as surrounding concentric rings of stromal and immune cells at defined distances from the tumor. And for each of these four different areas, we were able to perform whole transcriptome sequencing from regions containing as few as 50 to 100 cells. Here are some example studies that you can do once you have these whole transcriptome data in hand. If you look at tumor epithelial areas, you can identify driver signaling pathways, and you can look at transcriptional subtype. Are tumors predominantly basal or classical? If you use data from the stromal and immune rings, you can perform immune cell deconvolution and also stromal fibroblast deconvolution. But some of the analyses are more interesting when you start to combine those data. And if you take epithelial and stromal and immune areas, you can start to do things like paracrine signaling analysis, where you look for ligands that are expressed by one cellular compartment and then look for their cognate receptors and downstream targets in another cell population. And this allows you to start inferring how these different cell types are probably talking to each other in the microenvironment. The bottom of this slide shows results from a pilot study we did before we analyzed the trial specimens. And in this study, we took 4 tumors that we knew the transcriptional subtype of and identified 7 to 8 regions of interest per tumor and then segmented each of those into separate areas of illumination. And the t-SNE plot, as you can see here, is really broken down by 2 major axes of kind of separation for these AOIs that make sense. The first, which is shown on the horizontal axis, is a basal versus classical tumor subtype. You can see that of the 4 tumors we have here, those really span this whole basal classical axis, but each of the AOIs from each of those tumors clusters more tightly with itself than with the other tumor types, showing that the different AOIs are more similar to each other than tumor 1 is to tumor 2, for example. The other major axis of differentiation here separates out the stromal and immune areas, which you can see in blue and green up at the top of the plot, and then the tumor epithelial areas on the bottom. And interestingly, even though the tumors cluster very discretely, the immune and stromal rings are much more of a smear and are more continuously distributed across the basal classical axis. One thing that is interesting to note, however, is that the stromal and immune areas from T1, which is our most basal tumor, also tend to occur on the same side of axis 1, suggesting that there are differences in the stromal and immune areas in the basal tumor, even though basalness itself is actually defined by the epithelial cells and not by the surrounding tumor microenvironment. Once we had completed our pilot studies, we next turned to analyzing specimens from the trial. These results show what we found across our first 40 metastatic biopsies, including 26 pretreatment and 14 on-treatment biopsies. The UMAP on the left shows the same analysis breaking out tumor epithelium and then stromal and immune rings, and showing that, again, there are two major axes that tend to differentiate these, a basal classical axis and then also an epithelial versus stromal immune axis. One of the first things we wanted to ask is whether VDR agonism is actually changing signaling within these different compartments. And fortunately, a lot is known about the downstream transcriptional targets of vitamin D signaling, and so we picked out some of these and started to look at them. In the plot in the middle of the slide here, you can see expression levels for CYP24A1. This is one of the best known targets that is induced by active vitamin D signaling. And when we looked in pre versus on treatment biopsies in both tumor epithelial areas and also stromal and immune areas, we found that the expression of this target went up in both of those compartments, suggesting that vitamin D signaling is operative in multiple different cell types within these tumors. We found similar results for CDKN1A, which is also a known target that's induced by VDR, suggesting activity both in epithelium and in stromal and immune rings. And then conversely, we know that MYC, which is repressed by active vitamin D signaling, actually went down in both of those compartments as well. So this really gave us some interesting hints that adding vitamin D to chemotherapy for patients with metastatic pancreatic cancer was actually doing something to modify the microenvironment of the tumors in these patients. Beyond looking at our different cell compartments individually, we are also able to start looking at how those different compartments might be interacting with each other. And this is an example of an analysis from one case where we've looked at potential signaling mechanisms that are operating between tumor epithelium, as you can see in green, and then the adjacent ring 1, which is closest to tumor cells, shown in red, and then ring 2, shown in blue, which is further away. And in the circular plot on the left, you can see that there are multiple connections between each of these three different areas, although some of the strongest connections are between the tumor epithelium and ring 2 itself. And when you take those data and plot them in a different way, as you can see on the right, you can start to infer pathways that would be paracrine signaling between two different cellular compartments, or autocrine signaling, which would be that compartment signaling back on itself. And doing these types of analyses and others then allow us to understand how these cells are actually interacting, and for example, in the vitamin D trial, which cells might be primarily responsible for sensing a vitamin D signal that then might be causing secondary effects in other portions of the tumor microenvironment. And based on these analyses and other observations that we've made, we've started really delving into how the tumor cells in the microenvironment are interacting with the surrounding fibroblasts to set up the organization of fibroblasts in pancreatic cancer. And in this particular analysis, which we did by multiplex immunofluorescence, we used our fibroblast subtyping panel, which contains 4 different fibroblast markers, as well as markers to exclude non-fibroblasts in the stroma, along with a collapsed version of our tumor cell subtyping panel that allows us to distinguish basal versus classical tumor cells. And you can see that these two cell types often coexist within the tumor epithelium, as shown here in the cyan and also the pink or purple cells that are often in mixed populations within individual tumor glands. When we merged these data together, we asked a question about whether the fibroblasts within and that are surrounding the basal versus classical cells, whether those are organized in any different way. And in the plot on the left, we're looking at the probability of finding different major fibroblast subtypes as a function of distance from tumor cells that have a basal composition. And as you can see in the left-hand portion of the plot, each of the lines that represents a different fibroblast subtype are very separated, especially at the region around 25 microns. And so this indicates that these different fibroblast subtypes each have different probabilities of being at 25 microns away from basal tumor cells. In other words, if you're a basal tumor cell, you're much more likely to have a fibroblast that's in the gray line next to you than a fibroblast that's in the black line. In contrast, if we look at fibroblast organization as a function of distance from classical tumor cells, as you can see on the right, there really is no organization here at all. Each of our 5 major fibroblast subtypes are equally likely to be found at essentially any distance from classical tumor cells. And so this provides a hint then that it may be the tumor cells themselves and their polarization in terms of being more basal or classical that is actually driving and setting up the organization of the fibroblasts in the microenvironment that surrounds the glands. Finally, for the last couple slides, I wanted to show some examples of how we're able to integrate human and mouse studies to go deeper into functional testing of hypotheses that we derive from looking across large numbers of human pancreatic cancer specimens, most of which are only a single point in time, or if we're lucky, 2 different points in time from a patient in a clinical trial. As one example of this, I wanted to show some data from one of our collaborative studies studying a KRAS inhibitor using a mouse pancreatic cancer model. As many of you know, there are already some KRAS inhibitors targeting G12C that are approved for treating patients with lung cancer, and there are many KRAS and pan-RAS inhibitors in clinical trials at this point. And for pancreatic cancer, the approval of any of these agents is going to, I think, really change the landscape of targeted therapy for this tumor type, where more than 90% of patients harbor KRAS mutations. So in this particular study, we used MRTX1133, which is a non-covalent inhibitor of the G12D mutant allele of KRAS. And this potently inhibits KRAS G12D, but not the wild-type form of KRAS. And in this study, we treated the KPC mouse model, which has an activating KRAS G12D mutation as well as a p53 point mutation, and is one of the best studied mouse models of pancreatic cancer. And for this study, we treated these mice either with a short-term course of MRTX1133 or a long-term course, and attempted to identify mechanisms of resistance that allowed tumors, once they had initially responded and shrunk in the mutant mice, to then overcome being treated with the drug and to then regrow and ultimately kill the mice. And so when these mice ultimately died from their disease, tissue was taken from the mice and used for a broad variety of analytic approaches. And for what I'll show you today, I'll focus on the spatial profiling data. One of the biggest challenges of working with mouse models of pancreatic cancer is that, especially in the earlier stages of cancer development in these mice, the tissue in their pancreas is very heterogeneous. As you can see on the left, areas of invasive PDAC often coexist with areas of pre-invasive pancreatic intraepithelial neoplasia, or PANIN. And this is also true with areas of normal pancreas that often coexist with acinar to ductal metaplasia, which is the earliest step in the progression from normal to PANIN to invasive cancer. And these areas of normal and ADM can also exist with PANIN. To overcome this heterogeneity, which would cause significant problems if you did single cell or single nucleus sequencing out of an entire mouse pancreas, we again turned to spatial transcriptional profiling, where we could morphologically identify pure regions of either invasive cancer or PANIN, and then study the transcriptomes that are coming from the epithelial cells or the surrounding stromal and immune cells. And in the top set of images in this figure, you can see what our segmentation approach looks like for a mouse that was treated with vehicle in a region of invasive cancer. And we're able to very nicely separate out the cytokeratin positive or yellow tumor epithelial areas, and then the cytokeratin negative surrounding stromal and immune cells, even though this is a tumor that is very poorly differentiated and has really very narrow intervening regions of stromal and immune cells. At the bottom, here's the analogous segmentation approach for a region of PANIN, and you can see again that we can very nicely separate out the cytokeratin positive or yellow glands from the surrounding stroma, which is actually much more abundant in regions of PANIN versus invasive cancer. And here's an example of what some of the data look like from this segmentation approach. In the upper left, we're plotting about 200 areas of illumination across 8 mice, And as we expect, one of the major axes of differentiation here is whether the areas of illumination have come from epithelial regions or from stromal and immune areas. And as you can see in the upper right, when we then start to label those areas based on whether they were normal pancreas or pannin or ADM or pancreatic cancer, the other major axis of differentiation here is whether we have a normal or a precursor lesion or an invasive cancer, as you can see in purple on the bottom. And interestingly, if you look at the top clusters of dots, there seems to be a little bit of a spectrum or a progression as we go from normal to ADM and then over to pannin. And the plot at the bottom now colors these different areas based on whether the mice were not treated or treated with vehicle alone or treated with the KRAS G12D inhibitor. And interestingly, although we had really hoped to see some clustering here by treatment, that doesn't seem to be the case for these mice. And this has really suggested to us that the resistance mechanisms that develop in each mouse may actually be different, and that if you take all of the mice in the treated arm, for example, and pool them together, this may actually be diluting out different resistance mechanisms that arise separately within each mouse. And to get at those resistance mechanisms more specifically, we can take each of our areas of illumination and start to ask what changes are actually happening after various amounts or days of KRAS inhibition. And the plot that's shown in this slide shows genes that are enriched in either vehicle-treated tumor epithelium on the left, or genes that are enriched or that have higher expression in the KRAS G12D inhibitor-treated tumor epithelium. And this is kind of a sanity check from just the short-term treated mice, but many of the genes that we see that are downstream of KRAS are upregulated in the vehicle, as we would expect, as compared to short-term treated samples that got MRTX1133. In addition, in the vehicle-treated mice, pathways that are associated with KRAS, in addition to just individual genes, tend to be upregulated as well. And in contrast, when we look at the short-term treated samples compared with vehicles, multiple pathways are altered. And these include pathways associated with immune cells, such as monocytes, as well as fibroblasts and some stress-induced senescence pathways. EMT pathways are downregulated, and autophagy is also downregulated as well. And in pancreatic cancer, this is known to be a strong dependency of tumors that have activating alterations in KRAS. And so by doing these types of analyses, we're now starting to get a better handle on what resistance mechanisms are likely to arise when pancreatic cancer is treated with KRAS inhibition. This, of course, is in the mouse, but this allows us to predict what pathways might be altered in human specimens from patients that ultimately get this same drug. And so in summary today, we've talked about a number of points, and I hope I've been able to convince you that the value of doing spatial tissue profiling is really maximized when it builds upon existing morphologic and pathologic analysis. We don't have to look at tissue and take a kind of de novo approach. We already understand a lot of what's there, and we can use these exciting technologies to build upon that and see things that we can't otherwise see by eye and by traditional H&E staining. In many ways, spatial transcriptional profiling approaches are complementary to multiplex protein-based assays for tissue characterization. Each of these has strengths and limitations, but at the end of the day, I think it's important to note that we do live in a protein-based world, and it's very important to be able to validate findings that we get from RNA assays in protein space. It's also, I think, important to note that we can do protein-based and transcriptional tissue profiling at scale with high success rates. And we can do this in the context of human clinical trial specimens where oftentimes you only have 1 or 2 slides available for analysis. We can use these exact same approaches for human specimens and to do mouse studies as well and generate data sets that are very comparable. And perhaps most importantly, I hope I've been able to convince you that studying human tumors in their native environment in patients can actually improve cancer care. And this really allows us to do our thinking about how tumors are modified by either targeted agents or chemotherapy or immune modifying agents in people, and to understand how that biology is directly working in the context of clinical trials. And so with that, I'd just like to thank all of the folks in the lab that I run in conjunction with Brian Wolpen, along with the main collaborators we have, including the laboratory of Ande Aguirre at the Dana-Farber and those at other institutions shown here. If you have any further questions on this presentation, my contact information is at the lower right, and thank you all for joining today.
All right. Well, thank you so much, Dr. Nowak, for your informative presentation. Unfortunately, that is all the time we have for today, but before we close, I'd like to draw your attention to the Resource Center on the top right-hand side of your screen. Just click the links to open or download for further information regarding the presentation today. Now, this will conclude our session, so Dr. Nowak, do you have any final comments for our audience?
Yeah, thanks, Kaylee, and thanks to everyone again for joining today. I think the last two parting comments I'd really leave people with are that although we talked a lot about wet lab assays in this discussion today, analysis methods are really just as important as what you're doing in the lab, and maybe even more so. How you choose to define a cell and determine its phenotype, determine its boundaries, all have huge implications on the downstream data that you generate. I think that you could have the best assay in the lab, but if your analysis pipeline is not really set up well, if your QC isn't good, if you don't really understand the tissue structures you're looking at, you're never really going to get data that either makes sense or is reproducible. Conversely, if you have a good assay, maybe not the best assay in the lab, but something that's at least solid and a good analysis pipeline, you can still generate quality data that will be informative and that will be reproducible even in more sensitive assays. And I think that's just something that is often a little bit under-recognized in the rush to develop new assays and bring them on in the lab. And then I think the final point I would just make is that you really want to choose the best assay for the job. Every technology has limitations, whether it's plexing, resolution, sensitivity, throughput, or cost. All assays compromise somewhere, and there's not one single best choice for profiling either human or mouse tissue. It's really, I think, a decision that you need to make in the context of the specimens you have and the biological question that you're trying to answer. Certainly lots of exciting choices out there. The field is changing very quickly, which makes it exciting for us, but it's still important to have that degree of levity and pragmatism.
Great. Well, thank you again, Dr. Nowak, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.